All right, so we're becoming science vloggers for the night, and we're gonna isolate some cheek cell DNA. But first we have to do some prep work. So we're gonna make the lysis buffer and the protease solution. So we're gonna get 1.5 milliliters of dishwashing detergent and put it in a beaker.
There's one. Get out of there. There's 0.5 right there. Eh. Should be 1.5.
And 8.5 milliliters of distilled water.
Are these milliliters?
Yes.
8.5.
And 1/4 teaspoon of table salt.
Oh, you probably want this measured.
No.
No. It's just a big pinch of salt. That's all it is. Yeah. There's your deal. So what, now we mix it?
Yep. Stir gently until the solution is evenly mixed and the salt is dissolved. Gently. What part of gently?
I don't do gently. Here, we'll do it that way.
Babe.
What? I'm checking it. Salt looks dissolved to me. Oh, maybe not. Bubbles throw me off because there's soap in it.
I think that's why you're supposed to stir it gently.
So we're making the second lysis solution for second sample.
Where's
1 millimeter marks the top.
Got it. Well, not the very top.
No, it's top mark. Gotta be quick about it.
That's... Alright. And then 8.5 milliliters of distilled water.
Too much.
Okay, it's 8.5.
It's 8.5.
8.5. That's what I'm looking for.
Mm-hmm.
There we go. A half of a half of a teaspoon.
Half of a half.
Half of a half of a teaspoon.
Yep.
Of salt. Hopefully lavender dish soap doesn't mess up.
We'll find out, won't we?
Alright. So we are going to make the protease solution. And we're going to put 10 milliliters of distilled water.
Yeah. It's fine. Perfect.
Into the protease.
There you go.
And 1/8 of a teaspoon of meat tenderizer. And you stir it together until it's dissolved.
Gone.
Okay.
There you go.
10 milliliters.
Mm-hmm.
And
Regular pinch.
pinch.
Mm-hmm.
Of meat tenderizer. And it's dissolved. All right. And the first step of DNA isolation is, of course, to collect the DNA. So, my lovely assistant over here is putting 3 milliliters of distilled water into a cup. It's a cup.
Don't judge us.
And we're gonna collect some DNA. So now we have to chew on the inside of our cheeks for 30 seconds. Which is kind of a long time. Alright, let's speed this up. Okay, 30 seconds. Now we have to swish for 30 seconds. Okay, now we transfer that into the pipette.
Mm-hmm.
So the next part is to break open the cells, and we do that using the lysis solution, which contains detergent, and since the molecules are oil-based and hydrophobic, it will break those down. So I need 2 milliliters of the lysis solution.
Just use the thing on the side and add what you already have.
Or I can measure it out.
Okay.
1 milliliter. And 2 milliliters. And now we're going to invert this 5 times. Gently. 2, 3, 4, 5. There we go. So the next step after we break open the cells is going to be to get rid of the protein. And the protein stops the precipitation of the DNA. So a protease breaks down all the bonds and can destroy the proteins without affecting the DNA. Okay, so I'm going to get 100 microliters, which is right here. Put it
Did.
Okay, there was a bubble. All right, and we're gonna invert this 5 times. 3, 4, 5. And we let it set for 10 minutes. Okay, so it's been 10 minutes, and the next step is to add cold alcohol. It called for 95%, but I've got 91%, and it's been in my freezer because apparently during a disaster, everyone grabs all the alcohol they can. So hopefully that extra 4% doesn't cause any problem, but we're gonna try it. We'll make it work. You add it slowly to the solution and it separates. And you're gonna add up to 10 milliliters.
Not gonna be able to put the full 10 in. Apparently my mouth's got a lot of saliva in it, so.
What says up to?
We'll put it on 14 and leave it there.
So adding the cold alcohol very slowly down the side of the tube. And this allows the DNA to precipitate.
Some pretty wild stuff. It's right there.
Yeah.
It's wild. I think you got enough in there.
Let's not lose the pipette. We're gonna set that over there. And if you look, you can see that all of that's standing up in there.
Pretty crazy.
So now that we've done that, we're going to leave it to sit for 5 minutes before we come back to look at it again. Because of the high salt and the cold alcohol, the DNA that we extracted by breaking open the cell walls aggregates or comes together until it can be seen, apparently. So that's what we're going to wait for next, and we're going to come back and look at it. We already kind of saw the tall stringy bits, and what we're going to see apparently is it's thousands and thousands and thousands of strands of DNA, so it forms a fiber long enough that we can see it, because usually it's teeny tiny and microscopic. So this is cool. Alright, so it has been the 5 minutes. We've left it undisturbed, and look, this is pretty cool. So you have this really tall kind of stringy bit in the center there, if I can get it to focus. So we'll try to make it focus. You got a little in there.
Yeah, just one little strand. Where yours has like 6 or 7 different ones. Yeah. Or a bunch.
So that there's a difference.
I did mine wrong.
But now we mix it up, and then we're gonna leave it for another 5 minutes. So
can you shake it?
You can invert it.
Oh, okay.
Alright, and then we leave it for another 5 minutes.
Oh, now there's some.
Okay, so we've waited the last 5 minutes, and we're about to look at what's supposed to be our DNA. Okay, This one's mine. What's in there? Ooh, so all the white, stringy, clear stuff is DNA. Look at that.
Pretty cool.
Ooh. Oh, there we go. It's having a hard time focusing. All right, so we did it. We extracted DNA. We collected the cheek cells. We broke open those cell membranes with the detergent, and we broke down the proteins with the protease solution. And we made it insoluble with the salts and then visible by adding the alcohol. It was really cool. The stringy it kind of actually did look a little bit like a helix. It's got little spindly ends and very cool. Can't wait for the next